Welcome to our review on the eye. So the first thing we actually need to know then is what our eye is actually made up of. So in the table that you can see there, I've given you the name of the structure, a description of what it actually is, and what its function is. So you do have to know this, and probably the easiest way to do that is to put it on some flashcards, the actual structure on the first side, flip it over, and then have one for the description and another one for its function. So that means you can then just test yourself over and over again to make sure that you do know which part does what. So the first structure we need to know about then is the cornea. Now that's the transparent coating that you find on the very front of the eye itself. And the whole purpose of your cornea is to protect your eye and also begin to refract the light that enters the eyeball. Second part is the pupil, which is the central hole within the iris, the little black bit that you can see. And the whole purpose of that is to allow light to enter our eye. Third bit, the iris, that's the coloured part of your eye, and it's actually a ring of muscle. Now that's the bit that's going to change the size of the pupil by either contracting or relaxing. Next bit, the lens. This is actually a transparent biconvex lens, and the whole purpose of that is to focus light clearly onto the retina. Ciliary bodies, these are rings of muscle tissue that will alter the shape of our lens. The suspensory ligaments are, as the name suggests, ligament tissue that connects the ciliary muscle to the lens. And then finally, the optic nerve there is the nervous tissue that's going to carry those nerve impulses from the eyeball to the brain itself. The other thing they could ask you to do on the exam is to label or identify parts of the eyeball from a diagram. So the best thing you can do with this is to actually print off a copy of the eyeball, or if you don't have a print handy, just do an amazing sketch, because I'm sure you are all great artists. And then just have a little sort of post-it note or a little flap stuck over the actual labels so you can test yourself on what they actually are. So make sure that you do look carefully at where the lines actually go so you don't mislabel the pupil as the iris, for example. So if we actually consider what's going to happen for us to actually develop this image inside our brain of something we're looking at, the first thing that's going to happen is that as the light from the object hits our eyeball, then the cornea refracts that light to start to focus it. As the light then passes through the pupil, it then goes through the lens, which, if you remember, the whole purpose of that is to refract the light further. And the whole idea there is that it's going to create that sharp image on the retina at the back of our eyeball. As that sharp image is put on our retina, then the photoreceptors, which are those specialized cells within our retina itself, detect the stimulus and they produce this nervous impulse that travels down the optic nerve to our brain, which then interprets what it is we're actually looking at. One of the things we've got to remember is that when we're looking at things that are different distances from us, then our eye has to have some changes take place to allow us to focus on them. If you didn't have these changes, then when you looked at things that were different distances from your eyeball, some things would be blurry and only things at a very specific distance would actually be clear. So in order to focus on objects at different distances, the key thing that's going to happen is the lens changes shape. And the way we do that is as a result of the action of the ciliary muscle. So if the ciliary muscle contracts, then the lens becomes fatter or more convex. And that's going to focus on objects that are nearer to the eye. Whereas if our ciliary muscle relaxes, then the lens becomes less convex, so it becomes thinner, and that means we can focus on objects that are further away. And I've given you two diagrams at the bottom to show you what we're talking about. So on the left hand side you can see an object that's quite close to us, so if we're reading a book, for example, then what we can see at that point is that as it's coming through the lens there, it's a fatter lens. So that means that it's going to focus those rays of light on the back of the eyeball. Whereas if we compare that to the one on the right there, you can see that the lens is much thinner. So the reason for that is the result of the ciliary muscle either contracting or relaxing. Make sure you remember which way around it goes as to whether it's focusing on near objects or far objects. So we know what the eyeball should do, but it's not always perfect. So what we can find is that quite common issues, you've either got short sight or long sight. So anyone that wears glasses or contact lenses, you are the lucky winner of one of these defects in your eyes. So first of all, if we consider short-sightedness, this is where anything in the distance appears blurry, but things up close are absolutely fine. Now, the reason for this is that your lens is too strong or your eyeball is too long. So because of that, what we can see, if you look at the diagram on the left at the top there, is that the rays of light are actually meeting each other kind of in the middle of your eye. They're not meeting on the retina. So we end up with this blurred image as a result. In order to correct that, we need a concave lens. So the diagram below shows you what happens there. If we put a concave lens in, it bends those light rays slightly outwards before they enter the eye, and then when your lens refracts them back in, they now meet on the retina. So you've got a lovely clear image, and then everything is nice and focused for you. The second defect of our vision is long-sightedness. So this is where near objects appear blurry, but things in the distance are beautifully clear to you. So the reason for this is that your lens is too weak or your eyeball is too short. So you can see what happens in the diagram on the left at the top. As a result of that, what we find is those rays of light are actually going to focus in reality kind of behind the retina. So they're not actually meeting that perfect point on the retina, so we get a blurred image. In order to correct that, we use a convex lens, and as a result of that, you can see the diagram just beneath. Then it bends the rays of light inwards just before it enters the eye, and then as your lens further bends it inwards, we end up with those rays of light meeting perfectly on our retina, giving us that crystal clear image. The last type of vision defect that we can find is when people are colourblind. So if someone's colourblind, it just means they've got difficulty making out different colours. The most common form of colourblindness is red-green colourblindness, which is a genetically inherited condition. So it comes from your genes. It's not something you develop later in life. You'd be born with it. And it usually affects males. And to give you an idea of what it actually means for people who suffer with this, at the bottom I've given you an image. So on the top you can see the normal range of colours that we would see if we don't suffer with colour blindness. And then underneath you can see the effect of having this red-green colour blindness. So all of those colours there on that red side just have this kind of washed out colour. And you can see there's no real difference between the yellow and the green. So people struggle to tell the difference between those colours. So those people who are not colourblind, you look at them and see something completely different to someone who is red-green colourblind. The last thing we're going to consider is how we can actually see these things. So if we consider what makes up the retina, then we've got two types of photoreceptor cell. We've got the rod cells, which allow us to see in low light levels, but they're not responsive to different colours. And then we've got the cone cells, which are going to respond to the different colours. And what you've actually got are different cone cells that will respond to red light, to blue light, and to green light. Now, each of you will have a slightly different combination of the red, blues and greens, so that when you look at a colour, it's going to be slightly different to what your friend sees, just because of the combinations of the red, green and blue cone cells you have in your eyes. Obviously, that's just one of the natural ways that you will see a different colour. If you're a particularly mean older brother or sister, then you might have decided to tell your younger sibling when they were very small, pointing at a colour like the one on our banner there of the green and telling them, "That's blue." Hopefully you haven't done that, but bear in mind that when you look at that green banner, the green you are seeing is not going to be the same as the green everyone else sees, because you've got a different number of red, blue and green cone cells to someone else. Hopefully at the end of this video you can now identify the main structures in the eye, you can describe their function and also describe some of the defects of vision and how we can correct them.